Good afternoon, everyone, and welcome to another Atoms seminar. Before we start, I would like to point out that this year the Atoms group is collaborating with the Laboratory of Complex System Engineering from UNICAM, led by Professor Luis Franco, to bring you renowned speakers covering topics related to thermodynamics in this seminar. Today, the Atoms seminar has the great pleasure of welcoming Professor Arthi Jayaraman. Professor Jayaraman received her bachelor degree in chemical engineering from Birla Institute of Technology and Science in Pilani, India in 2000. In 2006, she completed her PhD in chemical and biomolecular engineering from North Carolina State University, and from 2006 and 2008, conducted her postdoctoral research in the Department of Material Science and Engineering at the University of Illinois Urbana-Champaign. In 2008, she joined the faculty of the Department of Chemical and Biological Engineering at University of Colorado at Boulder and held the position of patent assistant professor. In 2013, she joined the faculty of the University of Delaware, where she is until now as associate professor of chemical and biomolecular engineering and material science and engineering. Professor Jayaraman serves on the editorial boards of the ACS Macromolecules and ACS Polymers journals. Her academic career has been recognized with multiple awards and honors, such as the Aisha Concept Division Young Investigator Award in 2013, the ACS PMSE Division Young Investigator in 2014, and the APS Fellowship in 2020. Her research expertise lies in development of theory and simulation techniques based on molecular models such as coarse-graining modeling and application of these techniques to study polymer functionalized nanoparticles and polymer nanocomposites, and to design macromolecular materials for biomedical applications. So once again, welcome, Professor Jayaraman, and thank you so much for being here.
Thank you so much, Felipe, and thank you to the organizers for this lovely invitation. I've seen some of the past Atoms seminars on YouTube and excited to join that group. As Felipe pointed out, I am a faculty member at University of Delaware. The title I have now is Centennial Term Professor. I've been full professor for about 3 years now. My primary appointment is in chemical engineering. I have a joint appointment in material science and engineering. I also have an affiliation with the Data Science Institute. Currently, since 2 years ago, I've been serving as director of an NSF-funded NRT graduate traineeship program. This is something very near and dear to my heart. The topic of this traineeship that we run at University of Delaware is computing and data science, tools that my group uses, obviously, and connecting those tools to solving problems in soft materials. So the group of students in this traineeship are very interdisciplinary. They come from different disciplines. They learn about each other's disciplines. So this is something very near and dear to my heart. As Felipe pointed out, I'm also an editor for 2 polymer-focused journals, both based in ACS. One is Macromolecules, the other is ACS Polymers Gold. So if you're interested, I'm happy to talk about that as well in the Q&A part. My focus, obviously, today in the talk is on research in my lab and one particular area of research that we've been focusing some time on. So for those of you who don't know what my lab does, and I know that this is going to be posted on YouTube as well, so maybe others will see it and they don't know what we do. So a little bit of introduction to what we do and how we study things, and then I'll get into that one story that I want to share today. So my lab at University of Delaware and in the past at Colorado also has focused on studying soft materials. And when I say soft materials, I'm talking about materials that have polymers, colloids, soft colloids, mixtures of polymers and particles. And the polymers themselves can be synthetic polymers. You'd never put that in any biological system, or polymers that came from biology, came from nature, or are being made to be used in biomedical applications. So The pictures here are all from different projects we've had over the years. Some are active, some are past. And there are many more pictures that should be here that aren't here from more recent work. So just to give you an overview of the types of things we look at and the types of questions we ask. So my group has spent a good amount of time, I would say almost a decade, focusing on this top corner here, looking into designing polymers and using that design of the polymers to drive the thermodynamics, and then through that, drive the morphology of the system that contains both the particles and the polymers. So this particular picture here is a perfect one. It's called a polymer grafted particle. You may have seen in literature. Many groups study that. Our focus in my group had been, as I said, the hero of the story there was polymers, how we can design the polymer, the chemical heterogeneity, physical heterogeneity, architecture, chemistry. How can we tune that so we can achieve what we want to achieve with the nanoparticles in terms of structure, because that's what gave rise to the various applications where these materials are used. On this corner here, these are examples of projects in my lab where we focused on going from one type of polymer architecture to another. And so here's an example of a linear polymer, which is the canonical. When people think of polymers, they think of linear chains, but we worked so closely with really talented synthetic labs that have made so many advances that they can control exactly the types of architectures they can get. So how does changing a polymer from linear to, say, comb-like or brush-like, or from linear to cyclic to star, how does that change the thermodynamics of that system, not just the energetics, but primarily the entropic driving forces, right? Because you may have the same entities chemically in both chains, but one is arranged differently compared to the other, allowing for different conformations. So this is really cool thermodynamics here. And I know this group here is interested in that. We are also obviously interested in seeing how that thermodynamics then drives various structures. Some assembled structures are useful in applications, some assembled structures are useless. How can we get the good ones, and how can we not waste time, or at least the experimentalists not waste time trying to make things that are not of as much use? In the bottom are examples of systems where we've taken inspiration from biology to create materials for applications in medicine. So for example, here, collagen-like peptides and elastin-like peptides. So inspired by what we find in our body and making them with synthetic peptides or polypeptides, really using the sequence and the composition of these materials to control the phase behavior. Where do they transition from being soluble to insoluble and then soluble again? So those types of things are useful in drug delivery. Biocompatible materials. So these might be polymers that don't come from biology, but are compatible for applications in medicine. So polymers that are charged for gene delivery or complexation with DNA and then delivery of that. Or bio-derived, like polysaccharides. So here again, we are interested in looking at new chemistries derived from cellulose that give us the phase behavior we care about. I'll show an example of this problem in my talk in the context of the subject I'll focus on. So these are all the types of questions we ask. We always start with a question and then develop the appropriate tools in my group. These are the main tools we use. Really a big part of my group's expertise is in this top part. When you're thinking about simulating polymers, these are macromolecules, we often don't have the luxury to work with atomistic simulation. So we have to come up with the right type of coarse grain model for the problem at hand. We don't believe the same coarse grain model works for every problem. It's really question dependent. So a good amount of time is spent on this, and this is something we really enjoy. And then, of course, we use those models in simulations, molecular dynamics, Langevin simulations, Brownian, Monte Carlo, sometimes a mix of both. Sometimes we also use liquid state theory, in particular polymer reference interaction site model, inspired by the time when I was a postdoc with Ken Schweitz at Urbana-Champaign. And many times for the same problem, we use both techniques because then one has advantages in one way and another is advantageous in another way. In the last 5 years, a big part of my group is also pushing towards using machine learning, largely as a very powerful complement to many of these other techniques, and also as a complement to the experiments that happen in our collaborators' labs. So in short, usually a problem gets one or two of these methods to really solve that. And we are really very, very fortunate to have fabulous experimental collaborators with whom I work. very closely. My students work very closely. Postdocs work closely. I'll remember to highlight them in every part where I'm going to share with you their experimental results that we then analyze with some techniques that I'll discuss today. So as we've been working with experimentalists, we've come to really also appreciate areas where they could use computational tool development to help them analyze their experimental results and in turn allow for better connection to our computational results. So that's really been a big push in the last few years, I would say in the last 4 to 5 years, and we've really enjoyed that. So I thought I would highlight today that effort in my group. There's a lot of other things we are working on. I hope you'll look at my students' hard work in the publications through what we share in our group page. But my focus today is going to be on the method that we've been developing in the last 3 to 4 years and how we've created many case studies of how to use that method when you have experimental data focused on structural information. Okay, so the name of the method is CREASE Computational Reverse Engineering Analysis of Scattering Experiments. And really, this particular approach has flavors from all these tools. But today, I'll be focusing a little bit more on how machine learning has helped us and just highlight briefly where simulations can come in. I know many of you come from simulations background, so you'll appreciate the role you can play within this method as well. And of course, I'll highlight the experiments as we go along. Before I tell you why we needed this method, I should introduce the need for such a method. And what is the state of the art? Because I don't know if all of you are aware of these methods. Some of you may be experimentalists, some of you may be computational folks. So just very briefly, I'm not obviously the expert to do the scattering experiments. I give this information to you as I see it. So small angle scattering is one of the many scattering techniques that many experimentalists use to really understand structure in a material. When I'm talking about materials, I'm interested in soft materials. So these materials don't have crystalline order. There's a lot of disorder in the system, and yet they are very useful, right? Many of the formulations coming from industry, toothpaste, soap, shampoos are all formulation soft materials that don't have perfectly ordered structure, but structures that are still important. Those are the types of materials I'm interested in, my collaborators are interested in. So when they do small angle scattering, essentially they are subjecting this material that they don't know what the structure is subjecting it to a wave. And that wave, the input wave essentially goes through this material, and based on the chemical composition and physical arrangement, the wave is scattered. And that scattered wave is essentially captured here as the result. So this 2D scattering profile essentially has a lot of information in it. And the 2D profile is often used when you're looking at systems that may have some directional order, or it's not isotropic inside the material. But many of the examples in the beginning I'll show you are all systems that do have isotropic structure in them. And again, I want to be clear, these are not crystalline. There may be some crystallinity here and there, but the majority is what we call amorphous structures, could be ordered, but amorphous. They are often analyzed using this type of profile. So let me give you here an example from a paper recently, but this is not our paper, it's just from the literature. And this is the type of result that our collaborators would get when they look at this 1D scattering profile. And let me explain why this is such a good technique. Unlike microscopy, where you take a picture of that particular material at that time, static, at that length scale resolution, unlike that, scattering has a lot of information at a variety of length scales. This is inverse length scale. And so based on the intensity versus this wave vector, inverse distance, you're able to get information at multiple scales, and by doing so, understand organization of the molecules at different length scales, which is very useful. But the curve looks like this, right? It essentially looks like this. How do you know what is inside the solution if you look at this curve? So traditionally, the interpretation of these has been using analytical models, and analytical models exist for many canonical structures: spherical micelles, cylindrical micelles, elliptical micelles, flexible cylinders, etc. So those models have been developed for known structures and standard structures and make approximations. These analytical models are equations. You've written out terms making approximations. But sometimes it's not good enough, or there isn't even a good enough, close enough analytical model when you're dealing with structures that are not conventional, but intentionally obtained through really clever processing techniques, Or these are polymers that are making canonical structures, but the polymer is really not at all conventional and doesn't follow simple statistics like Gaussian conformations. So in those cases, there's a need for computational techniques. So we are not basically relying on inaccurate or very approximate analytical models. So that's really where the need arose. And it came out of actually a project that we were working on with collaborators that were making really cool new polymers, and the analytical model fits to scattering just didn't make sense. So that's when this method was born. So I'll give you a bit of an overview first of the method for those of you who are computationally leaning, and then I'm going to give you case studies of how we used it. And in many cases, I'll compare against analytical models to show where it does as well, where it does better, just so you can appreciate the strengths and at the same time, similar limitations or strengths as analytical model. So the main idea and the first step is what I'll focus on the most is when the main idea of the method is to take in as input either a curve, an intensity curve, or a bunch of curves from multiple small angle scattering measurements, X-ray neutrons, and really optimize through an optimization method. We choose a genetic algorithm, and I'll highlight why this is really useful. Basically optimize, find structures whose dimensions give rise to a computed scattering that matches experimental scattering. So in this case, I'm giving an example of in schematics of a vesicle structure, cross-section of that. And here you have, you're interested, as you will see why, I'll show you a little more detail of this particular system and why we are interested. But most people, when they're looking at vesicles, they're interested in the layer thicknesses and the dimension of that core, which is usually where they load drugs and other cargo. So the dimensions are usually of interest. So really the optimization of the genetic algorithm comes down to a family of structures whose dimensions give rise to a computed scattering that matches up with experimental scattering. That's really what we care about here. We're not thinking about molecules. We're not thinking about chains. We are really interested in structural dimensions. And note the bold, italicized, emphasized S. There isn't just one structure corresponding to a scattering profile. You'll often have a family of structures, and that's really what we like genetic algorithm. It gives us that. Second step, for those of you who are molecular simulation people, if you work with experimentalists, you can give one more piece of information and say, of those assembled dimensions, which ones make sense? Because some can be numerically right, but not physically right. You can go through with molecular modeling and simulations and fill in molecular detail into those structures and say how these molecules are organizing and what gives rise to that structure. This is also useful, like I said, to eliminate unphysical structures. There's just no way molecules that you said are in the solution are ever going to pack into these structures. So that's why the second step is useful, although I'll focus a lot more on step one today and highlight a few places, step two. So let's go into step one a little bit more into detail. And so, as I said, we are using genetic algorithm. You may come up with another optimization technique. We wanted to make it an accessible method for many experimentalists who may not be that computationally savvy. So it was important that we used a popular method. So the idea is we first have to get some input from the experimentalists. And if you're an experimentalist, this is what you need to give to your computational colleague. What is in your solution? This becomes important because we need to later on do the step two of filling in molecules, but it also gives us some additional pieces of information I'll highlight. Some idea of the shape, if you know from microscopy what the shapes look like, this looks like a sphere, it looks like a fibril, that downsizes the optimization to those structures and that's the dimensions. But if you don't know the shape technically, shape could also be one of the parameters you're optimizing for. Input of the scattering profile and information of whether you did it from X-ray scattering, neutron scattering, whether you contrast matched or not, because then that tells us which components of your system are what are scattering and the rest of it matches the background. This becomes important as I'll show you in a couple of slides. This is the information we need. Then in the genetic algorithm, we start with a population of individuals, which in our case are these structures, showing vesicles here, but you can think of whatever structure you're interested in. And these individuals are defined by their genes, which we say are the parameters we'd be interested in. So there are no molecules, there are no structures. We are really interested in these structural parameters. and essentially start with an initial population that's very diverse with a range of values for these. And then over time, we will optimize towards the structures whose IComp, computed IFQ, matches experimental IFQ. So in that initial stage, we have to calculate the computed IFQ for each of these structures. I'll go into that in the next slide. Compare that to experiments, and good match means good fitness for that individual, poor match, poor fitness. And has the fitness converged? So early on, obviously, the structures are so diverse, the fitness hasn't converged. Later on, over iterations, we'll come down to a state where most of the individuals' matches are not getting any better, so the fitness is converged. Just as any optimization technique, that's essentially the same flow here. So early on, it's not converged, obviously. So then you select your next generation based on good individuals, some crossovers, some mutations to keep the diversity and not getting stuck in a local optimum, but rather we need the global, right? So this allows for that. Very standard genetic algorithm. Many of you may be very comfortable with this method. And the output, like I said, is not one value for these parameters, but rather multiple structures that are in that final optimized generation, multiple structures, and so values and error bars. So that gives you dispersity as well, which is so much the case in soft materials. Things aren't perfect in molecular weight. There's always a distribution. There's always a distribution of structure. So this is really valuable for the soft materials people, much more than those who have crystalline perfect dimensions. So now coming to that point where I was making about calculation of the IComp, I'm just going to go ahead and put both of these. So we have two approaches. We start with the slow approach. I'm going to hide the fast approach right now. Slow approach is where for each individual, once we know the dimension, for that individual, the genes are its dimensions. In that, we place scatterers. So these are point scatterers. These are not molecules. These are not polymer chains. And we put scatterers only in those regions which the experimentalist told us is scattering, right? So maybe everything is scattering, so that looks like this. Maybe it's only the red zone is scattering because everything else is contrast matched. Then in that case, we put only the red zone, dots the red zone. Go through all the pairwise Debye correlations and essentially calculate the IComp through this very slow procedure. This calculation can be made fast through optimized code, yet it's slow in the context of the next method that I show you. So when we do this, we collect a lot of information of these parameters and its corresponding IComp. So we can really use that information after we've done that for a diverse set of initial structures. We can start to use that to train the machine learning model to take over from the next round or in the next run of the genetic algorithm. So the idea is these genes, which in this case is these dimensions, can be directly related to IComp through, in our case, we use neural networks. You may come up with another machine learning method, but essentially the idea is this is significantly faster. We're talking minutes versus hours on the left, minutes for the machine learning. So either way, we go through this iteration, whether we use the slow or whether we use the fast, the loop is identical. So that's the main method, right? Step 2, as I said, is essentially molecular simulations. I'll highlight that. So in the next 25, I'm going a bit slower than I thought. Next 25 minutes, I will share with you case studies of how we've used this method against experiments, and in some cases against our own in silico experimental data. What I mean by that, I'll describe in just a second. Here's the first example. Since I've been discussing vesicles, might as well go into that detail. The reason we got interested in vesicles is because of the work from Christy Kick's lab. Christy Kick's lab makes these nice elastin-like peptide, collagen-like peptide macromolecule. This is a collagen-like peptide triple helix. These are 3 ELP chains. They have really fabulous phase behavior. We worked with her in earlier papers to go after molecular driving forces. Not going to focus on that today. But when they come through certain transitions, we're able to collapse the ELP domain and self-assemble these into structures. Vesicles is one of those structures, and she uses those vesicles for drug delivery. And that's something we also look into in collaboration with her. In this case, we are interested right now in the dimensions of these layers and also in knowing how many CLPs are in the inner layer versus outer layer. And of course, the dimensions of the inner core. Well, what you'll see here is, so that's what we want. So those become the genes in our case. And we also see that the dimensions are not uniform. They're very folks will say polydisperse. Some communities will say polydisperse is a wrong word, but basically there's a distribution of domains. How about that? So as I said, the genes are the dimensions, and the genes are other pieces of information you may be interested in, which in our case was how many CLPs are inside versus outside. This is our goal. The scattering experiments have some challenges and are taking a bit longer. So I'm going to demonstrate in this particular case how we show CREASE works versus analytical model using data we made. So we knew that the most important thing for us to capture well in CREASE was these dispersities in dimensions. The vesicles, especially the core, is really has a very broad distribution. So how do we prove CREASE is doing a good job? And then how does it do versus analytical model? So I'm going to compare here with the analytical model, but just very briefly, I'll show you something that we do for almost every case study first before even going to experiments. We create our own IX, and I put quotes because essentially this is an in silico input. The reason we do that is then we know the answers. We can directly compare and say, did CREASE do a good job in getting the right answers? Of course, CREASE doesn't know this when it takes the input of the scattering profile. So when CREASE takes the input as the scattering profile, and then we also input that into the SASVIEW package with this analytical model, what do we get? What we find is, so here's the target values of the dimensions that we are interested in. The vesicle is just an addition of these dimensions. And so there's obviously an error bar because of the dispersities that we put in the input, in the calculation of the IX. And then I want you to focus on the genetic algorithm with dispersity. If you're interested, you can ask me what's the difference between these two in terms of implementation. I'll tell you that in just a bit. Machine learning allowed us to get into GA with dispersity. So you can see those boxes are pretty spot on top of the target value, especially compared to the analytical model fits that are often off. So in this case, when the vesicles are very dispersed in dimensions, that's where CREASE really comes to be useful, and especially in comparison with the most popular vesicle analytical model. I will say this, we also did this with monodispersed vesicles, and in that case, both the analytical model and CREASE do identically well. So this becomes really valuable when systems lead to dispersity in vesicles, which might not happen in all soft material cases, but does in our case. Just very briefly to highlight why step 2 is useful. Like I said, you can eliminate some of the dimensions of saying this is absolutely unphysical, or you can say, I would like to understand how molecules are packing within these layers. So if you have models, atomistic or coarse-grained or Martini, I know you had a talk on Martini not very long ago, you can fill into those dimensions the molecules to see how stable they are and how they pack. We had a model already because of our past work, a coarse-grained model for ELP-CLP, and so we essentially were able to recreate a slice of the vesicle so we can see concentration profiles and then see how many CLPs are on the inside versus outside conformations of the ELP. So molecular reconstruction can give you pieces of information that you're not getting from step 1, regardless of whether you used analytical models or you used CREASE GA. So this might be of use to many of you. Now I'm going to go into a second example of systems that are fibrillar in nature. So let me actually introduce this problem. This is a polymer synthesized in Karen Woolley's lab and characterized in Darren Pochan's lab. And the information they had looked like this in microscopy. So we know it's not a sphere, not a vesicle, not a spherical micelle. It's fibrils. It's something. We can see one dimensions. And then we have the scattering profile as well. But these polymers are very new. They have not been characterized in terms of persistence, lengths, et cetera. So this falls in the family of the polymers that led to CREASE anyway. So this was almost the second study we did after that first JAX paper that led to the birth of CREASE. So the question that they wanted to ask was, look, if you look at the microscopy, you know they are fibrils, but you have no idea what the cross-section is. So what's the cross-section? Second question is the cross-section of this type core corona, meaning it's phase separated by hydrophobic hydrophilic, or is it just more uniformly packed, just arranging themselves into whatever fibril with whichever cross-section? So these are the types of questions you may pose. And in some of these cases, we did have analytical models for fibrillar structures, but I'm going to show you how we used CREASE to test hypotheses. This is not the most attractive slide, but I'll walk you through it. dimensions, and you can see the types of dimensions, i.e. the genes that you would optimize in the genetic algorithm. And depending on the type of structure you're interested in, cross-section, circle, rectangle, ellipse, core corona, or if you do the other one where everything's uniform, your dimensions are going to change. But we wanted to first check if they were going to form core corona. So we have corresponding microscopy data from Darren's group. So that only gives that one dimension, which is the width of the fibril, nothing about the cross-section. And then the cylinder models, these are all in analytical models in SASVIEW package. And so when you fit that, you get the 2 dimensions, right? What you're interested in first is to check if the core corona model makes sense. So we tried that in CREASE. There is none in analytical models. So when we did that, we tried different scenarios where they can have independent widths. And none of these even come close to the real dimensions. When we tested the second hypothesized structure, that they are more uniformly packed, then those dimensions are more in line with the dimensions that we see in microscopy as well as in the analytical models. So here's an example of a case where CREASE was more useful to test hypothesis. Even though there are analytical models out there, they may not be there for all scenarios you want to test. So this is one example. I say 2B because I'm going to give you another example of fibrils, but these have different genes or different parameters that we are interested in. And so let me just very briefly tell you about this material because this is an ongoing project. We're really fascinated by this. So the solution that we are going to study is methylcellulose solutions. Cellulose is basically this structure here with R's all being H. So these are all OH groups in cellulose, which makes cellulose really hydrogen bond beautifully to create a crystalline structure. As you probably figured out, I'm not a fan of crystalline structures. I'm more interested in amorphous structures. So methylcellulose is a great example of where you replace some of these H's with CH3, so hydroxy with methoxy. And because of that, your hydrogen bonding is disrupted. So the material is more soluble in water. So it's not crystalline. It's essentially a solubilized state. Or the beauty of this is this structure, because you can play with temperature effects on hydrophobicity and hydrogen bonding, it's a solution at low temperatures, and at higher temperatures, it self-assembles to form a network structure, fibrillar network structure. So there's much interest in this system in industry because these are great food additives, and various pharmaceutical personal care products can come out of this. So there is a very close interest in various communities on how is the structure formed, how are the chains packing. And there's obviously you can also go away from commercial MC. When I say commercial MC, it basically means how many of these 3 are replaced by methoxy. On average, it's 1.8 among the 3, but you can also change that. So there's a lot of interest there. Okay, so why am I bringing this up in the context of CREASE? So there's a lot of experimental data from the University of Minnesota groups. I see Ilya was in the participant list, so Ilya's colleagues. They have very nice scattering data that they were so willing to share. We got that data, and we really wanted to analyze that to see if some of the conclusions they got by using analytical model, when we did that with CREASE, do we get the same? And then we went one step further and said, hey, can we also understand why chains pack the way they pack? And so that's the second step. So this is ongoing work. I don't want to show this just yet because we're finalizing a manuscript and that will be submitted very soon, but I'm going to tell you the one that we have published. So as I said, Tim Lodge's group, Frank Bates and coworkers had a lot of data of methylcellulose solutions at various concentrations, various chain lengths, beautiful scattering data that they gave to us. They themselves had done the analytical model fits in the publications, and they used flexible cylinder model. And then they had basically analyzed the parameters they got to take out, which is the average diameter. And they found that the diameter, interestingly, regardless of molecular weight, regardless of concentration, is always in a certain range of 17 to 20, right? So this was surprising because many times you would think if you change the molecular weight, if you change the concentration, you may change the entropic and enthalpic balance, and you could get bigger fibrils, smaller, but in Swiss quite consistent. So is this because of the analytical model that it couldn't resolve that? And if we were able to take a lot of the dispersity into account, do we still get this answer? So that's really what our first task that we had. And this would also be a nice test on CREASE for a system of great interest to a lot of people. So again, very fast now, because I think you understand the flow. Essentially, we had to decide on the genes. So in this case, our genes were flexibility parameter, Kuhn length, diameter, and essentially their distribution. So the dispersity in both of them. And so exactly like I described before, we go through the various steps of the optimization and in the end get a bunch of structures, all of whom have good computing scattering match with experiments, and then they basically are distributions of both of these values. I'm focusing on the diameter here, so there's going to be a lot of data. I'm going to quickly walk you through this. Again, the data for the experiments came from Schmidt et al. Paper, specifically among the many papers that those groups have written. I'm showing you only the data for 0.3 weight percent. We have the other ones as well, which behave similarly. The first data point I'm showing is the diameter and the error that came from analytical model fitting. This is the average value of the diameter for various molecular weights, and you can see they are consistent. Then when we use the machine learning based CREASE, that's neural networks, the NN, so neural network based GA, we always do multiple runs just in case we get stuck in a local minima. We want to make sure that's not the case. And indeed, it's not. We get consistent results, and they are consistent for molecular weights. They are also consistent among the various concentrations. So this to us was a good validation, not only of the science, but also of the fact that our genetic algorithm, our neural network-based algorithms giving the same answer. I can tell you, this is the part I've shown you, I can tell you based on the results we've gotten that we are writing up, essentially the draft is almost there, is that we do see if we started from coarse grain models of methylcellulose and watch them assemble, they actually do form consistent diameters. And there's a little extra twist in that story. You have to just wait for the paper, but it's a consistent picture there as well. And we've done both atomistic and coarse grain. So you just have to wait for that. And I'm sorry that I'm going to switch to the next case study. So before I switch to the next case study, I want to highlight something specific here, which is that I've been focusing on solutions where the concentrations are low. What that means is the materials are assembling themselves into various structures, micelles or fibrils, but those micelles, vesicles, fibrils, they're not talking to each other. So they're far apart. So the structure factor is really one. What we call form factors is P of Q. Some people will just use the word structure factor for everything, but it's important that we highlight the difference. To us, this is that elementary particle, which in our case so far have been these different assembled structures, fibrils, micelles, vesicles. But most scenarios are not lightly concentrated. It's more like this, right? Where you have forms that themselves have dispersity in dimensions, and they're going to talk to each other because of high concentration. So structure factor is important. What people tend to do in these situations is they usually do the experiment first at a very low concentration, iron out the P of Q, then they assume, which in most cases might be okay, but not all cases, that P of Q doesn't change just because things got concentrated. And then they use the P of Q that they calculated from low concentration at the high and focus here only on the S of Q. But just as I explained to you, that may not be the case. As things get crowded, they may reorganize, so the form factor might change. So CREASE has to be able to handle both. So again, let's go through this step by step. First step, let's try to go after S of Q when we know P of Q and we know the P of Q won't change. So now we're talking about mostly hard particles. Just because they're crowding, they're not going to change their shape. So we had a project where we were interested in mixtures of melanin, it's a bio-inspired, our skin material, melanin mimics, which we made out of, not we, our collaborators made out of polydopamine materials, and these became hard particles. Mixtures of melanin and silica. So here in this project, these are hard particles, and the reason we're interested in them is because when you mix them together, you're able to control color, as you can imagine. That's why we're using melanin. And the particular things about the structure that give rise to the color are the degree of mixing of these particles and the mixture composition. You may say, well, why don't they know the mixture composition? Well, when they're doing the experiment, they start with a mixture composition, but the final assembled structures can have more of one particle than the other. So they don't know the mixture composition in the assembled particle state. So these are important because we have shown experimentally and computationally that the degree of mixing of the two particles and the composition gives rise to a spectrum of colors. So Of course, if you change the chemistry and didn't have silica or didn't have melanin, you could get other colors. But the same melanin silica, when arranged in different ways, gives rise to different colors. So that's the property we're interested in, coming from the structure, coming from the design, just essentially these particles. So how did we handle CREASE? How did CREASE handle this? So just as I said, in this case, these are hard particles. So let's do it the way experimentalists do it. So our collaborators, Ali Dinojwala, materials came from Nathan Gianeschi's lab. They did the low concentration experiment, used a very well-known analytical model, spherical form factor, which should work fine. It did, and confirmed that these particles are spheres. So P of Q is known. These particles are spheres with dispersity. That's important. So P of Q is known. So now let's do the experiment at high concentration. That's what they did. Beautiful scattering results came from their lab. So notice that these are assembled particles. This is the supra ball that is made of these melanin silica particles. So they do the IFQ on these supra balls. And you can see the experiments are the yellow dots. First, they did the traditional model fit with a well-known analytical model that many would use. That's the green line, red line here. Not a great fit, but this is what most people would use. But if you use the same scattering and put it to CREASE and let it solve, and then give out the final answer of the structures, those final structures, computed scattering, sits spot on. Look at the black line and the yellow, the gray line and the blue. You can't even see the gray line because it's right under the blue. So it did a better fit. But that's only one part of the story. You can have a great fit, but have a really bad structure, right? So there has to be one more level of proof that the structure is right. So what if we take that reconstructed structure that comes out at the end of CREASE after you've done the optimization, do a color calculation on that structure. If that structure's color, calculated color, matches with the experimental color for that same system, then we have a confirmation that the structural features, i.e. the degree of mixing and composition, are right. So that's what we did. So the experiments in Dinojwala lab gave the scattering. They also did the color response. Then that scattering comes to CREASE. CREASE does the optimization. We have a final structure, optical modeling on it, computational response can be compared together. So this is obviously the reason I'm showing you this is there was a match. Here's the color chart, the experiment computation matchup. You can also see reflectance profile for the experiment and computation matchup. These are different compositions of melanin and silica. So this is a confirmation that the structural optimization was done right, and the structure, representative structure of those parameters is also correct. So this is one particular study. Obviously, I showed you the case where the P of Q was not changing, and I kept motivating that the P of Q and S of Q have to be solved simultaneously. So our most recent study with results, experimental results from Bhuvnesh Bharti's lab at Louisiana State, was a system where he was looking at mice cells that when you made them crowded, the mice cells, well, in his case, it's a hard core, but a soft shell, and that soft shell evolves as your concentration increases. So he can't use the P of Q from low concentration to high, so he brought the problem to us. I know I was given 40 minutes to talk. I'm already at 40. So I'm obviously not going to tell you everything about the experimental data. I encourage you to look. This is an open access paper. Most of this work is in open access journals, so everybody should have access to this. But very briefly, the whole idea here is now those genes or those parameters have both information about structure, like degree of mixing, concentrations, compositions, and form, shell thickness, particle radius, and you're simultaneously solving for both of those. And then that in the end, the output is essentially information about that assembled particle. So the shell on top of the particle, nanoparticle surfactant shell on top of the nanoparticle, as well as their arrangement at that condition. So this was a great use in his experiments because analytical models were just not working for him. So I want to highlight that these are just a few case studies. We've been working very hard and showing to the community with experimental data how CREASE works. And so we have a code out there, but we really want people to take away the philosophy rather than say, should I use that code? Because I think computational people should be able to work with experimentalists to take this philosophy to those experimental measurements. So it's really the workflow that we are hoping you'll borrow, and then you can write your own fancy optimized code. But the whole idea that I want you to leave with, because some of you may say, well, this is different from reverse Monte Carlo. I want to highlight, unlike reverse Monte Carlo, where you evolve a structure, here we are optimizing the lower dimensional representation of that structure through parameters, using machine learning to make the connection between that lower dimensional parameters and computed scattering. And the third thing is, in the end, we're not getting one optimized structure, we're getting an entire family of optimized structures, and that's very useful in materials like soft materials. So this is really what I wanted to mainly focus on. But some of you may be machine learning gurus out there and say, well, why genetic algorithm when there are fancier machine learning methods? As I said, we wanted it to be an accessible method for our experimental collaborators. But Chazao Liu in my lab recently found beautiful data online in Science Advances with an open source database of structural analysis. And in this case, Chazao handled 2D scattering and developed a method that very nicely can take 2D scattering and predict the microscopy image, simultaneously a microscopy image into 2D scattering. But in the context of today's talk, what this method basically does is it takes, I'm going to go to the next slide. It takes as input scattering, which is a harder to interpret, right? That's what we've been struggling. That's why we had the CREASE method, and converts that through these autoencoders, but in particular, it's a variational autoencoder, and essentially predicts it into a form that we can easily understand, right? A microscopy image is so much easier to visualize and understand, there are these structures in comparison to scattering. But of course, you can do this either way. Some people use this to get should use this for bottleneck characterization from high throughput. Some can, like I said, use it to take harder to interpret, convert to easier to interpret. And this can be very useful in downstream other applications for property, etc. So I'll wrap things up here by thanking the people who did the work. I'm just a mouthpiece in a way, sharing with you sort of years of hard work. My other team members do a lot of other cool things too. Unfortunately, we don't have the time. But this method started with Daniel Beltran-Villegas, who's now at Janssen, Johnson & Johnson's offshoot company. Mikhail Vessels is at Siemens. Christian Heil just graduated recently, went to Dow. Current members who are wrapping up, ZJ Wu, beautiful methylcellulose work that I showed you, vesicle work that I showed you. Stephen's a newer student who's taking over slowly some of these, and newer members who are contributing to the newest set of data we're getting from companies. And of course, my collaborators who gave us the data to test our methods, supercomputing, and funding sources for various projects, because these were all not one project, they were different projects. But thank you for your attention.